Now, folks, I'd like to welcome two people to the podium to talk with us. First of all, I'd like to introduce Professor Michelle Waycott. So Michelle is Chief Botanist with the State Herbarium of South Australia. Her research is focused on Australia's botanical biodiversity and its effective management. Michelle leads two major components of the Healthy Coorong Basin Trials and Investigations project, so we will be hearing again from Michelle later today. But the two areas she leads are Component 2, Aquatic Plant Condition and Future Options for Macro and Microbiota, and Component 7, Science Integration Across Trials and Investigations components. Do you want to commence first and then I'll introduce our next speaker? Will that work? Thanks, Michelle.
Good morning, everybody. It's really great to be here again, seeing many of you. Thank you for participating in these community engagement activities and hearing our latest. The work that I'm presenting today is focused around the primary producers of the southern Coorong, and I'm going to talk about the aquatic plants and algae, and then Tamar's going to talk in a few minutes about the microbiota. I just wanted to put a big shout out to the team who worked on the aquatic plants and algae component of this work. It's a big team, as you can see, lots of names, and it was very collaborative and extremely hardworking and dedicated team. And now I'd just like to also recognise some of the community members who helped us out. Jeff in particular, with his incredible support with drones and equipment and gear, and his always never-ending enthusiasm for telling us what's going on. One of the things I like to get people to think about when we're talking about the primary producers of the Coorong is that it's very much an issue of scale. We often think about what's happening at very small local scales, but in fact the system operates at this scale, the bigger scale. And so we have to trade off our understanding of, from an ecological perspective, of what's happening almost at a metre squared scale to how that influences and changes the system as a whole. I just wanted to sort of remind us that the baseline from which we're sort of thinking about what the system's been doing is reflected in this graph here, where the Ruppia community essentially disappeared from the system during the Millennium Drought, and it started to come back but went into kind of this oscillating cycle for a few years of not being particularly consistent. But by the time this project started in 2019, we had the beginnings of a more consistent distribution of the species. Now, at the time when I started working on this system in 2015 in a lot of detail, it was thought that Ruppia tuberosa was it. Well, we now know, yes, Ruppia tuberosa is a dominant part of the southern Coorong, but there's a range of other species that occur there. In particular, another species of aquatic macrophyte, Althenia cylindrocarpa, or for those of you geeks out there, Lepelina cylindrocarpa. Plus, there's another species of Ruppia that is not Ruppia megacarpa. It may in fact be a new species to the Coorong. But what this tells us is there's more than just one plant that can cope with this system, giving us a little bit of hope for the future, and we're starting to see some of that with these floodwaters where we're seeing other species start to recover. We know a little bit more about that because we did some tricky environmental DNA work that shows us that, the dark blue dots are the presence of Ruppia tuberosa across our sites from, this is plotted against distance from the Murray mouth, but we also see Althenia, another species of Ruppia, and a couple of different forms of Ruppia tuberosa, again, emphasising a bit of diversity. The biomass of the system tells us that by the end of a major survey in 2021, which was sort of early summer, that there was moderate build-up of biomass across a large range of the system. So the bigger the orange dot, the more biomass in that general location. And what this tells us is that the plant community following the drought had really started to recover to essentially the distribution pre-drought and some of the biomass pre-drought. And this is summarised a little bit better here for considering the variation between seasons. So in 2020, the summer of 2020, there was more biomass closer to the Murray Mouth, sort of around Noonamina and in between the Needles and Parnuka Point. But by 2021, that biomass had shifted a bit further south. The conditions in the southern end of the North Lagoon weren't as good. And then if you look at the right-hand plot here, this is where the plants grew over a longer part of the year. So what this is telling us, if we look at it, is the plants come and go and they're variable in space and time. And it's an important lesson for how we sample and how we look at what's going on in the system. You have to sample widely and you have to sample at the right time. What are the environmental conditions that are good for the Rupia community? And I'm going to call it the Rupia community as all of those species all growing together because they all do co-occur. The conditions on this top plot here, we've got biomass against temperature, which is a bit of a surrogate for the change in the environment over the seasons. The blue bars there on the left telling us that at the end of the growing season of 2020, which is into the summer, the plants sort of did their standard die-off as the water levels drop and the salinity goes up, the plants die off. And then we have the community growing up to a more reasonable biomass again at the end of the year. That seasonal cycle's normal and typical. Not what we saw this year, by the way. Water depth, there's a depth range around which the plants are found occupying the system. Kind of what we expected, but we've got some good numbers on this now. In the 2020 season, we saw it between plus or minus 0.4 AHD. So that's the sediment height compared to ocean sea level. That's the standard numbers you see people refer to depth. But what that tells us is the main populations of the community occur in this strip. That's that water level. And that's the target water levels that Adrienne's team try to keep the water up in the later part of the year to maintain these communities. So this gives us some really nice numbers for that. What was really interesting was in 2021, we saw the populations occurring in deeper water. And that's probably the consequences of the La Nina conditions, and we saw the plants seeming to grow a little bit more perennially. So that's between years. The salinity differences between years also did something really interesting and quite important to observe. We saw the biomass change again. The peak biomass is around our 60 parts per thousand. Either side of that, it can drop off. But what we saw in 2021 was we saw a massive shift to the plants forming turions. And when you measure biomass, what that means is the biomass lasts longer during the year. And so there was a big difference between those 2 years. We also can tell from the tide height data, the periods where the water levels, so the shaded areas, this plot's a bit back to front, you shouldn't look in the middle, you should look at either side. The plots on either side, you want the water levels in the grey zone to be between the green bar. And so the further you are away from that, like over on the right here, like happened in 2021, we saw the conditions not be as good for the plants. And we can map these conditions over time, and Claire later on is going to talk about how we use this kind of data in modelling. But what we can do then is track back through time and see how well the conditions prevail for the plants. And these shaded colours I've put on each one is how well the plants would grow under those conditions. So red is not so good, orange is a little bit okay, and what we're really after is the green zone. So we now know enough about the plant's growth to try to aim for some particular ranges of conditions. Sediment condition, as Luke pointed out, is really important. This sediment RAP score is basically, if it's a low number, it's terrible. If it's a high number, it's pretty good. And what we see is we only get high biomass when the sediment condition is good. Now, whether that's a chicken or an egg, we don't quite know yet. There is definitely a conditioning effect of the plants on the local sediment, but it also might be that when the sediment conditions are poor, the plants find it hard to recruit, in particular, into those areas. The seed banks are, again, aligned essentially where the plants grow. Makes sense, but we now know what those depth ranges are, and some of those seed banks are now getting pretty healthy. So long term, for resilience, we want a seed bank over 2,000. What this tells us is most of the time we're not getting that still, and that's even after all that time since the drought. This has let us clarify the life cycles of the plants that we're working with, and we've now done some detailed analysis of the phenology. And what this tells us is when you go out there every couple of weeks and you look at what the plants are doing, we know that the plants do that build up to maximum growth in late spring, early summer, and then they do their die off as the conditions change. Now, this is some work we did linking what's happening with the nutrients in the system with the plants, and these are plant tissue nutrient ratios. And this little grey box in the bottom left is where the tissue nutrient ratios would be suggesting that the plants would be nitrogen limited. In fact, they're not nitrogen limited because there's so much nitrogen in the system, they don't know what to do with it. And that's exactly that process that Luke was talking about. There's excess nitrogen in the system. The seagrass plants, the aquatic macrophytes can't take it up enough to bind it, and so it's floating around and available. What's really interesting is that the triangles are a whole different slope, a whole different relationship, and that tells me that from work we did in 2016 to work we did in 2020-21, there's been a fairly big shift in the system, and not in a great way, much more nitrogen. Relating all of that to how we would interpret the system in terms of the relationship between nutrients and plant condition. Essentially, again, echoing Luke's summary, we get better performance in terms of biomass and the number of turions, the number of structures that the better quality sediments give us. So sediment is key, and essentially lower nutrients are also key because you want the lower organic content and lower percent ends. Now, the filamentous algae in the system is a symptom. Now, it's natural to have algae growing in these systems, absolutely natural. But when there's so much of it, it's a sign the system's unhealthy, and these can manifest themselves as these big mats that form in any time from throughout spring and summer, and they bind up the plants, they inhibit flowering and seed set, and we saw direct relationships with those. And what we did was we started to measure the amount of algae being produced, and the work is ongoing to work out what the relationship between the new high water levels and this algal biomass are. But the consequence of it to the system are these organic, oozy, sludgy sediments that Luke spoke about earlier, and that brings with it a whole range of different microbial communities and processes. But what we found was the production of this algal community followed a life cycle. And this life cycle is really important to consider. The algae don't just sort of appear out of nowhere any time. They actually appear following a trajectory that matches the trajectory of the aquatic macrophyte. It's the Rupia community. And it's a fairly predictable relationship. We also found that this algal community caused light stress at a significant level. At Parker Point and at, North McGrath Flats, there were periods where this, the light stress was so high that the plants would have been physiologically and, demonstrably affected by the lower light. All of this together has given us a really clear idea of the changes we need to make to try to get back our desired Rupia community, and that the target for that community has to be about restoring better sediment quality, having nutrients available in the right form, and so that the plants can take them up. And also that the community can be made up of more species. So we're picking up 3 out there now, but in the most recent surveys and some of the observations that Faith Coleman's made and Glenn Hill as well, commenting on the presence of other seagrasses, on the presence of muskgrass, the charophytes, Lamprothamnium, and these are historically been part of this community that we'd essentially lost, and we're starting to pick them up in our samples as well. So what this is saying is we can move forward and look at what's going to happen in the future. Thank you.
Thanks very much for that, Michelle. I'd now like to invite Dr. Tamar Jamieson from Flinders University to talk with us some more. Tamar is a postdoctoral scientist at Flinders University, where her research focuses on biofilms in the marine environment and biofouling management strategies across maritime platforms. Tamar has been investigating the decline of aquatic plants in the Coorong with a focus on understanding the microbes that are involved, as well as exploring ways to restore the aquatic plants and ecological balance of the Coorong. Welcome, Tamar.
All right, thanks for coming everyone today. This is my first community event and it's a pleasure to see you all here. So today I'm going to speak to you about the microbes and their relationship to the Rupia community. So I guess you all know this would be the desired outcome when we have inflow into the Coorong, where everything lives in a very balanced system, where everyone gets along, shares nutrients, and the Rupia grasses prosper. Unfortunately, due to lack of flow, we end up sometimes with this kind of system where the balances get out of whack. So then it's survival of the fittest, and those which adapt to the changes quicker, they survive better. But unfortunately, you get the algal mat production, which then reduces the ability of the Rupia grass to grow. As a microbiologist, I consider microbes very important. I think that's the downfall of my degree. But anyway, I think they're important to indicate system health. As one of the smallest organisms within the water, they feel the impact of changes quicker than, say, a fish or an invertebrate. So they can really be important to ecosystem health and the management thereof. But they're also really important to the seagrasses. As you can see here on leaves of the seagrasses that we took from the Coorong, you have a very nice community living on the leaves. And this is really mutually beneficial to both the plant as well as the microbes. And we found that they were really important to the survival of the plants as well, as they have this relationship where the microbes will produce certain chemicals that the plant requires, but the plant also provides chemicals and nutrients to the bacteria. Just give a small snapshot to the study that we undertook in the Coorong with Michelle Waycott's team. So we had 5 sampling sites along the Coorong. We had 2 in the South Lagoon, Wild Dog Islands and Policeman Point, 2 in the Central Lagoon, Parkner Point and North McGrath Flat, and 1 in the North Lagoon, which was Noonamina. And we undertook 4 sampling periods, which kind of coincided with the seasons. In October, December, March and June. Overall, when we looked at our sample collections, we could see how the environmental conditions really impacted our samples. So you can see here in the spring and summer were different from the winter, and this was mainly due to pH and ammonium, where in autumn, the autumn samples were different from all the rest of them due to temperature and turbidity. So seasonality definitely has an impact on the microbial community within the Coorong. So then we considered, well, maybe these impacts are on the sample types. So when we identified the bacteria within the samples, we can really see that they're really independent of each other. So the water samples produced this type of community where there's dominant organisms which are seen in the larger circles, but then the smaller circles which are equally as important, but maybe not so many within the sample. But they were also very different from the sediment samples, as well as the leaves of the Rupia sample and the roots. So these communities are quite mixed and very independent of each other. We then considered how these were influenced by each other, and we had a look at which ones contribute to the differences in each of our samples. So the blue samples here are our water samples, the brown were our sediment samples, and the green ones are our Rupia samples, so leaves and roots combined. And we can really see that the microbes that were in the samples which generate the differences are quite independent of each other. So they're not influencing each other in any way. When we took that down to Rupia level and we considered the different life stages of the Rupia, where we have turions and rhizome presence in the blue, in the browns, which when only the roots were present, or in the life stage where the leaves and roots were present, we were looking really at, because the Rupia is in the sediment, maybe the sediment has some bearing on the microbes that you do see on these plants. And we found that the sediment really did have an impact at the different life stages. So the sediment provides the plants with different microbes that are available, and it is healthy for the plant. So if the sediment is important where it grows in, maybe the roots are equally as important, is what we considered next, as in, because they are in the sediment. So when we looked at the community on the different life stages, we could see also that they were very specific to the life stages, those communities which were on the roots. They were very selective, but they still created this very diverse and specific community. So while they were independent, and they were all linked to these sediments, the question we really asked was, why these ones? Why were they so important to these Rupia plants? So we took into consideration the functions or the capabilities of the bacteria that were on the plants. So maybe they perform something specific that the plant likes. So this is a heat map showing all the different functions that the bacteria do. And as you can see, they're quite sample site and sample date independent. So at a specific time, there's no real pattern therein. So we can see here that if we focus on the yellow ones, which is the highest amount of bacteria that undertake these samples, we can see that there's 3 or 4 functions there which are quite high. And these functions are specifically functions where the microbes break down chemicals that either they find in the water or they find on the plant, and then makes it into something else. So this we determined was quite important for the plant, so clearly they needed these bacteria to perform these function, which really implies that the Rupia plant is selective of its bacteria based on what functions it can perform for it and to help it grow in a healthy system. So really I would just, I guess, like to get you to take away is that microbes matter, even if you don't think they do, and that they're really important for the Rupia plant as it develops in the Coorong. I would like to acknowledge the students from the Le Terme lab that assisted with these field trips, as well as Emma from the Michelle Waycott lab, even though she's not there anymore, as well as funding that we received from the Goita and Department of Environment and Water for this project. Thank you.